mean, occasionally you get hybrids among species. In one extreme, they're either non-viable or they're mules, who can't themselves reproduce. But even if a hybrid can reproduce, if it reproduces much less efficiently, I would say those two entities are different species. But knowing where those boundaries are can be difficult. So defining those boundaries does have conservation implications. And, I'm always mixed about some of these things that are going on where people want to de-extinct organisms by reconstructing their genomes, because I think we could do more good.
I saw Jurassic Park, man. I am not going.
Yes.
All right, we're just talking with my good friend here. We're having a great time, but I want to introduce him. This is Edward L. Braun. Is that good?
Yep.
Wonderful human being, cool guy, evolutionary biology down there at the University of Florida, among other things. You do molecular evolution, stuff like this. You're dealing with numbers, right?
I'm really a computational guy, a numbers guy. I do have a lab where I can have people isolate DNA. I've periodically walked in there. I'm not doing it a lot. I mostly get A's, C's, G's, and T's sent to me by my colleagues in a computer file, and I don't actually touch any real organisms or anything real. I touch the keyboard of my computer.
But you like your animals. I think you've been around the world as well and saw some wonderful places.
Been around, obviously since I'm a computational person. I'm more actually sort of a tourist and visiting colleagues to sort of talk to people about problems and things like that. But yeah, I've had a number of wonderful collaborations in China. Unfortunately, have not been in China since, the pandemic happened.
Sure.
Although I hope to go back again. It's a wonderful place. I've been everywhere from the far north, right up close to the border with Russia and North Korea,
wow,
to as far south as the island of Hainan.
That is so cool. So what was your takeaway from that? Not many people get to make that trip. Was there big differences in the set? What did you feel? Was it relatively safe? I think the infrastructure is pretty good over there, right?
Oh yeah. No, I mean, I tend to when I've gone to China, other than the fact that I can't read anything.
Sure.
Actually, I always learn like a few symbols that are really important, but it's just like being in America or Europe, really. I mean, other than, everybody's Chinese.
Yeah. That's the big difference you notice right away. Yeah.
Yeah.
But the more I see of it, the more I think, well, there's a beautiful landscape over there. And then they're building infrastructure too that is just massive and really interesting.
Oh yeah, very much. I mean, I've stayed near the Olympic Park from, was it the 2004 or 2008 Olympics, I believe.
Mm-hmm.
And that's just an absolutely beautiful stadium and stuff. I mean, that infrastructure is just incredible.
Yeah.
But, really, I go over there and, I have colleagues I talk to. I, have colleagues in, across the U.S., some in Europe as well. And, I wish I could say that, I had great insights going out and actually being collecting organisms and stuff. But um, I'm mostly useless in the field.
Right.
I'm sort of like a tourist. I'm like, okay, I enjoy looking at stuff, but don't
Just
expect
bring me
any
back the data and I'll look at it. I'll be in my
exact.
office. No, there's nothing wrong with that. Yeah.
Yeah, no.
But other than that, I mean, you get to go to China. You get around. Have you had? We were just talking before this about some of your wow moments and in some of the work that you've been working on, and you're basically trying to map out the genomes of certain things. So what all are we considering, and what are some of the applications when we get down to that molecular level and we start taking this out and putting this in? What's kind of the end goal?
So really, um, if you think back to like the 19th century when folks like Darwin were sailing around the world on the Beagle and stuff, and various other naturalists were learning about nature. That's when we named a lot of organisms, at least non-microorganism organisms. And I would say the 21st century is really when genomics and the technologies for genome sequencing have reached a point where we can do the same thing, but at the level of genes. So instead of going around the world naming things, trying to understand how they fit in with each other in a natural history sense, we can now do genomic biodiversity.
Okay.
And, that actually has, I'm interested just from the philosophical standpoint of, well, how do organisms work? Going all the way down to the molecular level, but it can have real world implications in the sense of, conservation implications, being able to even rigorously define the units we want to conserve. Yeah. Because, at one level, a species is a group of organisms that exchange genes or alleles, versions of genes within the group and seldom outside the group. Occasionally you get hybrids among species. In one extreme, they're either non-viable or they're mules, who can't themselves reproduce. But even if a hybrid can reproduce, if it reproduces much less efficiently, I would say those two entities are different species. But knowing where those boundaries are can be difficult. So defining those boundaries does have conservation implications. And, I'm always mixed about some of these things that are going on where people want to de-extinct organisms by reconstructing their genomes, because I think we could do more good.
I saw Jurassic Park, man. I am not going.
Yes. We don't want to de-extinct any dinosaurs. Or I should say non-avian dinosaurs, because sure. Of course birds are dinosaurs, but most birds ain't that dangerous. Yeah. Although I will say one of those occasions where I went out in the field, I was helping somebody who was grabbing alligators.
Oh my God.
Which are surprisingly calm once you kind of restrain them a little bit and often put a towel over their head to kind of, keep the visual stimulation down. But one of the fish and wildlife people who was along helping us had also captured, what was this? I think it was think it was a rail. I'm now drawing a blank on exactly what bird they
Yeah.
grabbed. Um, but they said, "Oh, we would like some blood samples," and got more scratched up from the bird than from the alligator. Because the gators, once you get them under control, they're like, okay, cool.
Yeah.
I'll just
That's
go along with
Isn't that
it.
funny? Are they just they're just so confident? They go, well, and the bird is obviously always prey and it's just panicking like
Yeah.
crazy.
Well,
You got bad luck in the field, I think.
that was, get alligators. I mean, it was. I always joke about this because I always feel like, all the people who claim to be abducted by aliens, that we were doing alien abduction to these poor alligators because, I was helping these guys. We were out in the airboat. You shine a light across the lake, and you get a lot of eye shine from the alligators. So you can see these two eyes staring
Yeah.
back at you. And then they aim the airboat at it. And, one of the people who knows what they're doing lies down in the front of the bow of the airboat and just grabs the gator behind the head, pulls him out. And it's a very macho thing. I didn't
Right,
do
right.
the actual gator.
No, I like animals. It's like the catch and release thing. It's like you don't even want to eat the fish. You just want to make him late for something. He has to go apologize to somebody. Say, I'm sorry, I'm late.
Yeah, I mostly, other than my pets, I mostly leave animals alone myself.
Yeah.
And my pets, we have two dogs, a tortoise, and a turtle.
Oh shit.
And
What kind of dogs you got?
they're both rescue dogs, so sort of mixed breeds.
That's
One
the
is
best
a
kind.
cross.
Okay.
So he's, how they say a dachshund is half a dog high and two dogs long. He's a cross, so he's, three quarters of a dog high and a dog and a half long.
Not bad. Not making progress.
He's named Oliver for the Green Arrow, Oliver Queen.
Oh, cool.
So.
Yeah. No,
Yeah.
I love the mixed breeds too. What about the other one?
Harper is our other one, and I have no idea what she is,
Yeah.
other than about 40 pounds and very excited by walks.
Yeah.
She loves her walks.
How old are they?
So Oliver he just turned 9, and Harper is 3. So, and they're buds. They play and,
well, I'll send you my picture of mine when we get off, and you send me yours.
I should send you a photo of Harper and Oliver as well.
These are the best companions you're ever going to find.
Exactly.
Yeah. So what did you always had an interest in nature? Obviously, you're an animal lover. You got a turtle, you got a tortoise. When did you in your life first, look at nature and go, well, this is nice?
I think I always remember to some degree being interested in various aspects of nature. I always thought I was going to be a scientist, even at an early age. I wasn't really sure what kind of science. And, like most kids, I thought, I probably thought I was going to go in space or something. Who knows? But, you don't know what being a scientist is like as a kid, really. It's a lot of sitting in your office, even for people who go out in the field, just for people who want to do it. But it is a wonderful thing because, ultimately it's about saying, well, gee, how does the world work?
Yeah.
And, obviously my specific interest is in gene sequences and how genes work and how they change over evolutionary time. So, I've drilled down a lot into that, but
Well, where did that come from, though? Was there obviously you don't just 5 years old think about that and go, "Wow, that's what I'm going to be." There was maybe a moment that you went. When did you decide that? Oh, this is the little
well,
stuff.
yeah. So, I would say that if anything, um, probably when I was really young, I thought that, like I said, space was cool. And so if you had asked me, once I exceeded the age where my response would have been astronaut, I think my my response would have changed from astronaut to astronomer.
Ah,
And
right.
so sort of thinking about that. So even, like in high school and stuff, I kind of, was interested um, in sort of a mathematical approach. I had the hobby. I was a nerd in high school, believe it or not. I know you look at me and go, "Oh, this guy was never a nerd," but
Yeah.
okay, yeah. um, but I was into computers, so I thought, you know, these all fit together. And of course, as you're getting ready for college, you're starting to think practical. Is somebody going to pay me to just think about these things I find cool? And
Yeah.
in a moment of self doubt, you go, "Oh no, no way, nobody's ever going to pay me to do that." Um, I figured, oh, I'll be like a computer programmer. Surely they'll pay for that, which, yeah, they do. And I probably could be making more money on that. But then I took a biology course, and, although I'm old enough now that, sequencing and so forth was really, um, it was not something that was a something that really permeated down much to undergrad curriculum. But I started to see a little about that, and I went, I like computers. And this stuff seems really interesting. And maybe, all this stuff I dreamed about as a kid of, calculating velocities among galaxies and things, maybe this is the cool place to go instead. Instead of looking up, I'm going to look down at the DNA. Um, and then I kept doing that. So,
That's incredible. So how in the course of your career, how has the what do we know about the field has changed, and how has the equipment and everything changed?
oh, it's changed remarkably. In fact, a lot of things, there are things where I wish I had been more of a dreamer when I did this, because there are things that we do now that I would have considered science fiction when I was when I was an early graduate student and stuff, like easy genome sequencing and things. You know, uh, when I was in grad school and I started hearing about people wanting to sequence the whole genome of yeast, I was like, because my PhD work was mostly focused on budding yeast, which is brewer's yeast or baker's yeast,
Mm.
which is a wonderful model system for genetics. And it was the first eukaryotic organism, an organism with a nucleus, to be fully sequenced. But when that was going on, I was like, well, is it really worth it? I mean, why not figure out the function of a gene and then sequence it? And, 30 years later, it's sort of like, well, let's just sequence it and see what's in there, And I think nothing about, let's sequence a bird genome, which is, of course, the yeast genome is about 14 million base pairs, give or take.
Okay.
And a bird genome is about 1 billion base pairs.
Okay.
So, for reference, our haploid genome, I'm giving the haploid genome size. So most organisms, we're diploids. We have one copy of each chromosome from mom and one from dad, or in the case of a yeast, they don't have males and females. So if it's a diploid yeast, one from the alpha parent and one from the A parent. Uh-huh. But, the size of half our genome, our haploid genome, would be 3 billion-ish base pairs. So, now it's a huge amount of data, and just obtaining it is very inexpensive and fast. And I think it really revolutionizes everything, because we can look at the variation among organisms at the genetic level with this incredible high resolution. What are the bases that make up gene X? Gene X's.
Now, I'm going to start asking you stupid questions, because this is so intense. But so what's the smallest or like the most simplest creature that can be sequenced, has the least amount of, nuts and bolts to it, that still is a living thing?
Okay, so I guess that's a good question.
Is it?
And it's a wonderful question. So I guess it depends on what you mean by living thing, because if you mean like a bacterium that has its own metabolism as well as its own genome.
Yeah, sure.
We're talking, about 500 genes and, 500,000 base pairs. But, I will have to say that despite the fact that most of my research, especially recent research, has been focused on birds and to a lesser degree reptiles, I have a really soft spot in my heart for bacteriophages. And bacteriophages are viruses that infect bacteria.
Ooh.
And I actually I've taught a couple of times a course in bacteriophage genomics. It was really a computational biology course, but using bacteriophage genomes for that. And, you can get super small bacteriophages, sort of 3,000, 4,000 base pairs of, of RNA or DNA that can be enough that it can hijack a bacterium, make more copies of itself, and go on. I consider those kind of living.
That's pretty cool.
No metabolism, but
it's like giving the bacteria a little taste of its own medicine there.
Exactly. And I was even telling my genetics students yesterday that, if you want to really revolutionize the world, maybe you should take up, how do bacteria resist bacteriophage infection? And the reason for that is, biotechnology is largely based on our ability to cut up DNA in a way where we can plan where the cuts are. And the enzymes that allow you to do that were discovered from bacteriophage interactions. So, it may seem like, well, figuring out how bacteria resist bacteriophages. That's so far away from how something that seems practical, like, how do we resist bacteria or how do we resist viruses? But figuring that part out revolutionized biotechnology. And we're now in the CRISPR revolution, which is this technology for being able to rewrite DNA. And where did that come from? Bacteriophage interactions. So I've now talked myself into changing my research program right now.
That's what it's all about, yeah.
Joking aside, though, I really, it's sort of a love of mine that's not been something I've really been researching, but it is something I've integrated into my teaching and so forth. So,
yeah. Well, this is amazing. Bacteriophages, in the sense that something so simple can be so aggressive, and yet is it understudied still? Do people study it, or is there more we can know about it, or use it to cure disease?
So actually, that's a good question. And the answer is people were contemplating phage therapy for a while. And then, of course, we got antibiotics.
Sure. But that has
and of course, antibiotic resistance.
Uh-huh.
And there is some renewed interest in phage therapies.
Cool.
So, cool. Being able to, the big problem, of course, is, trying to figure out how to expose bacteria that are causing a disease to the phage. You're essentially giving the disease agent its own disease.
Right.
So, I actually have been contemplating some things that I might take this from a bit of a avocation to a vocation, because, well, viruses evolve quickly. We unfortunately have gone through 3 years of learning how quickly viruses can evolve. But being able to test the sort of computational and mathematical framework that I use to reconstruct the evolution of birds. I could do evolution in the lab, and this isn't a unique idea of mine. There are people who've done similar things in the past. But if you could essentially artificially create an evolutionary tree in the lab of phage, you might be able to then analyze those and see how good your methods are at figuring out the tree. So with the type of stuff I do, where I've been trying to estimate the evolutionary history of birds from genetic information, a lot of that action is tens or 20, 30, 60, 70 million years in the past. We can make inferences about how well the methods we're using behave, but we really don't have a direct way to go and test them. And that's frustrating. We have to go with consistency of results. And I think we can get far from that, but it sure would in some ways be nice to have evolution of a real organism where what the evolution was because you did it.
Right. Right.
You were growing it in the lab and allowing it to mutate.
And I would guess the real hope is that computer modeling will make it so that you can run 1,000 tests in 1 second and get to your conclusion.
Yeah. And we certainly, and I've done some of this too, which is to, you essentially rerun evolution in a computer.
Mm-hmm.
The challenge for that is you have to have some model for what the probabilities of different types of changes are.
Right.
So
Again, we talked about this before, the unseen, unknown elements that no one could
yes,
find. And that's why when climate modeling comes out and it says, well, it's not bad, but it's like there are domino effects probably that,
exactly. And, all of these models, and, there's a famous statistician, George Box, who has the phrase, all models are wrong, but some models are useful. And the thing I always tell students is, well, and the trick of being a good scientist is having ways to figure out whether a model is useful or not. Because if you say, oh, well, is the model wrong? Well, of course it is. Most processes in the real world are very difficult to capture computationally or mathematically.
Yeah.
But that's not the operative question. The question is, okay, have I built a model of this process that's good enough that can make predictions that are useful? And obviously that's a hard thing, because how do you know? Well, the reality is you have to sort of cross-check different types of data and so forth and say, oh, if this model predicts X, which I can observe on a short time scale or something, I'm more confident that it can predict Y, which might be on a longer time scale or something like that. So I mentioned before when we were talking about the fact that in birds, so what we ultimately want to get is like a family tree of birds. So there are 10,000 species of bird, and with a group of people, I have some grant support to generate that, family tree of all 10,000 species. So at the species level, if you look at different types of data sampled from a genome, you actually get different answers, which sounds scary.
Right.
And it is, because, you ultimately would expect that if you analyze enough data, you should converge on the same answer. And what really appears to be happening, and this was one of the things probably one of the papers I'm most excited by, was something we published in 2017. If you analyze protein coding genes, you tend to get one type of tree, one family tree for birds. And if you analyze non-coding data, you get a different type. And when I say type, what I mean is that you have these trees. They look sort of like family trees. A little different because you're starting from one ancestor, 100 million years in the past, and then it breaks into two species, which break into two species, and so forth. But ultimately, these mathematically are just graphs. It's possible to get a number that says how different is tree 1 from tree 2. So what we did in this 2017 paper is we collected together a bunch of trees calculated using coding data and a bunch of trees calculated using non-coding data. And we actually generated an additional tree using a non-coding data set that we generated for the paper. But we then calculate how different the trees are and then cluster them. essentially we're clustering them in a way that makes a tree of trees. So it's not even really a family tree. It's one level up. It's basically a tree that tells you how similar the family trees are to each other. And what we found was that all the coding trees were in one part of what I'll call tree space, and all the non-coding trees are in another part. So it was a very consistent. They were converging on the same answer. It's just that there were two answers, one for coding, one for non-coding. Now, the question is that real? And the answer to that is almost certainly no. There is probably one family tree for birds. So now we're left with which one's right. And the answer could be coding's right, non-coding is wrong. Non-coding could be right, coding could be wrong, or they could both be wrong. They can't both be right because, it's the same genome. And the trick, I would say, so spoiler alert, I think non-coding is right. And there's actually a reason for that, which is all of those trees were generated by comparing the same nucleotides in the same gene. So you take a bunch of genes, you identify the same gene in different organisms, you line them up in such a way that the same nucleotide, A, C, G, or T, lines up with the same nucleotide in a different organism. So there we're looking at changes from A, C, G, and T. You've got 4 letters of DNA, and how are they changing amongst each other? Another type of genomic change you can have is, there's a big class of things we call rare genomic changes, like a little bit of DNA flips around, so it's inverted. It's something called a microinversion. We did
is that from falling down and just getting a rock in your shoe or something? How do you get that?
Well, one of the people who goes out in the field a lot and collects birds with me, I was gonna, because he had collected a bunch of samples, was going to add him onto the paper I was writing on microinversions, because, of course, some of the data was generated using things. And he goes, you know what? I have no idea what a microinversion is, other than maybe it's like what happened when I fell down and I microinverted myself. So
I've been microinverted. It hurts.
Microinverted after, having enough scotch or something.
Right. Yeah.
But what these probably are, so, when we pass our chromosomes on to the next generation, or when a bird does it or whatever, the chromosomes recombine. So, you had the chromosomes one chromosome you got from mom, one you got from dad. And when you pass them on, they mix themselves up.
Right.
And these microinversions are probably related to some of the processes that go on for swapping the DNA.
Lost in transit.
Essentially, exactly. There's essentially sort of problems where you literally are marching along the sequence and a sequence is reversed, like flipped over. So those happen very rarely. They're also computationally hard to find. At some point, I keep telling myself I'm going to rewrite the code I wrote, 12 years ago or so to do this project and make it more efficient. Haven't yet. We did the initial publication. But a type of rare change that's easier to find are insertions and deletions of DNA. And the cool thing is we found enough insertions and deletions that we can build a family tree, not of nucleotide changes, not of asking, what is the pathway of change among A, C, G, and T, but instead, oh, here's a chunk of DNA that was deleted, here's a chunk that was inserted into the genome, et cetera, et cetera. And since that is probably happening under different rules, well, we have different biases from reconstructing nucleotides. I'm not making the assertion that none of them are biased, that any of them are unbiased. There might be biases hiding in any of these, but if you can get two really different ones to agree, they're probably telling the same story.
This sounds very similar to like playing pin the tail on the donkey in the dark.
Yeah, but a lot of science is like that. You sort of have to check your method. I mean,
sure. I mean,
what non-scientists are often don't really think about all the time is the degree to which how you're measuring something can affect the conclusions you come to. And how do you know anything? How do we know anything? Well, we cross-check among things. And in my opinion, at least, if you have two very different processes where you can go in and interrogate them, and how things happened were probably somewhat different, and yet, despite that difference, they kind of, come together to give the same result, and you sort of say, I think we're on to something, and maybe we're not perfect, but
yeah,
I think we're on to something.
Yeah, it's like, and even the stuff that's, etched in gold, we have to go back and say, well, let's take a look at this one more time, because, everything we know about soil and everything we know about Alzheimer's has changed drastically in just the last 5 years.
Yeah, no, exactly. You always have to reevaluate things, I periodically, there have been a few examples where I've tried to, in my teaching of genetics, sort of talked about, how we learn things. And I had a joke where I sort of brought in some comparative things from physics. And I just, because everybody knows Einstein, I sort of, made the joke that, people didn't know how this works. So, during 1905 in Einstein's miracle year, Einstein said, "Hold my beer." I'm going to tell you. Which, of course, the students laugh at and stuff like that. But, I still always emphasize that, what we think we know always has to be reevaluated. And what we know may only be an approximation of the truth. Right. But you know what? I mean, approximations of the truth are pretty good. I mean,
best we can do.
Yeah, no, exactly. I mean, if we waited until we knew everything, we'd still be living in caves. I'm not even sure if we'd be painting really nice, paintings on the wall, because we'd still be trying to figure out how to do the paintings right.
Right. Yeah. We've come a long way, baby. Yeah. Now, good. So we went over time. You're going to come back. You will chat again. I really like to have you back.
Loved.
Edward L. Braun, University of Florida, evolutionary biology, molecular biology, evolution, all around beautiful human being, brilliant man.
Thank you.
Anything you want to add before we go?
No, I'm not sure. Other than I really want to thank you for giving me the opportunity to do an interview in a series that also included John Waters. So
yeah,
I cannot thank you enough.
Yeah. That's how I get all the big names in academia. Just put John Waters out there. Yeah. Now he's a wonderful guy, class act, and I was grateful to have designed his Christmas card last year, and he turned around and used that image on his tour when he came up and
that is incredible. That is incredible.
called me on the phone out of the blue sky, and you that voice, you know that voice, and I was floored. But yeah, good guy. But all right, well, we'll sign off to these people. We'll talk after this.
Yep.
But thanks for coming, everybody. Bye.
Later, all.